It is 10 years since Luella Kenny last set foot inside her own front door. No one knows when it will be safe for the family to return. But tragically, there will be fewer of them. Biotechnology may be our best defense against the chemicals with which we poison our surroundings. There are other powerful inventions that could transform our lives, too. But Luella, like many people, says take care.
Exercise caution and make sure that we don't have it out in the market and suddenly somewhere down the line find we have a bigger catastrophe, such as we've seen in the use of DES or thalidomide in the past.
What would men born before the first industrial revolution have made of scenes like this? Work that would once have been done by 1,000 men can now be done by a handful. But there is a trade-off. The machines that help them cost millions of dollars, and the fuel they burn a king's ransom. Smelting the ore disgorged from thousands of giant tinker toys like this is a cause of acid rain. It's the story of modern industry in miniature, a story that billions of new workers are about to rewrite. According to the evidence of fossils, bacteria were the first inhabitants of Earth. They are everywhere, deep in the ocean, high in the atmosphere, in frozen wastes and boiling springs. We never see bacteria, though there are more of them on each of us than there are people on the planet. A few cause disease, but most are good to us, using their digestive juices, enzymes, to break down the world's garbage like a natural waste recycler. This is the chemistry biotechnology is learning to exploit. Bacteria don't stop at eating organic matter. In the New Mexico mine, water is sprinkled through huge dumps of ore containing worthless iron pyrite and valuable copper. Bacteria attack the pyrite and produce oxidized iron and sulfuric acid. These dissolve the copper, producing a lagoon of copper sulfate blue at the base of the leach dump. Because of its taste for iron pyrite, better known as fool's gold, Theobacillus ferrooxidans is coining real profits for the copper miners. The next step is to remove unwanted metals from the solution. Finally, the purified solution flows into an enormous bath. Electricity is passed through the liquid, and the copper is deposited on plates.
it's really a very nice tool for a mining property like ours to have available to it. It takes a very long time to recover copper out of a leach dump. That's one of the disadvantages. The traditional mining and milling methods and smelting methods are very quick turnaround on the copper, very quick recoveries. Leaching isn't quite as quick. However, we can make copper for about half the cost that the conventional method does.
As well as saving money, mining microbes save the environment. No smelting means no acid rain. Biotechnology promises to produce a cleaner tomorrow, but it may have ways to mop up past mistakes as well. The beauty and the power of Niagara Falls have captivated visitors for years. But the power it could generate has also lured industry to it. William Love started it off. The canal he began to dig in 1894, intending to found a metropolis on hydroelectricity, was abandoned after a mile. In 1978, this long-forgotten trench, which then had a school built over it, achieved a notoriety it will not live down. In the 40s, chemical waste had been buried in the canal in metal drums. In 1978, rotting drums rose up in a bizarre parody of a resurrection. The panic that followed revealed alarming levels of cancer, miscarriage, and birth defects in local residents who have since been evacuated. Now, Love Canal is a far cry from the pleasant suburb Luella Kenny moved to with her young family.
It was just idyllic, truly, with the oak trees in the backyards and the wandering down to the creek and just putting a fishing pole in there and just exploring for frogs and pollywogs and things that young boys would enjoy.
The Kenny family were not to know that the creek was polluted. Even after her youngest son, John, had a spell in hospital with a mystery disease, Luella did not suspect that chemical wastes from the Love Canal dump were poisoning his favorite playground.
He continued to play in the backyard and towards the creek. And then we found out later that the creek had 32 parts per billion of dioxin in the spot where he played. We even had an occasion where a bird went into the creek at that spot, drank the water, and dropped dead in front of the state officials. And the state officials were just trying to tell us stories like the bird was died of parasitic worm infestation. This was John's room. Our tinker toy is still here.
All through the summer of '78, John Kenny became more and more sick. But in or out of hospital, he never lost his sense of humor.
Still as sick as he was, because you could see he could hardly walk. His feet were swollen, his whole eyes and everything. And he wanted to tease his grandmother that he didn't come home, he was hiding. And I can remember him answering the phone and her asking him, "How are you, John?" And he said, "I'm fine." He says, "Nani." He called his grandmother Nani. He says, "I'm fine, Nani." He says, "Don't you worry about me because your blood pressure will go up and then you're going to get sick." But anyway, as it turned out, then he just started having difficulty breathing, and we took him back to the hospital again after 2 days, and then he just died.
there will be other love canals. You may live near one of them, hiding places for the industrial nightmares that are left over from yesterday's consumers' dreams. The all-electric homes of the 20s and 30s made great demands on the electricity supply industry. As well as more power, better distribution was needed. New equipment that wouldn't catch fire when surges occurred. The answer was a wonder of its day, a stabilizing fluid that kept its cool no matter what. And stayed, and stayed, and stayed.
That stability, which was carefully worked into the material, was also what made it so difficult to dispose of once it was no longer used or needed in the equipment. The PCBs, as they've come to be known in the last few years, have the kind of a structure that is very difficult for most organisms to attack.
Just how hazardous PCBs are is still a subject for debate. But no one's taking any chances. These dumps contain a witch's brew of suspect compounds. Dust to dust, ashes to ashes. Most things decompose. But 20th century technology has granted PCBs the gift of eternal life. Or almost. You can destroy them with great heat, but many countries don't have the facilities, which is why the biotech approach is causing such excitement. PCBs have been around for 60 years. Was that long enough for nature to respond? Were there any bacteria that could dent the chemical's armor? That is what scientists from General Electric set out to find on a contaminated site in New York.
And indeed, almost every place we looked, we did find bacteria that could grow. This flask contains bacteria that have grown on the parent compound of PCBs. We then took that culture and added some PCBs to it to assess the competence of each of these different cultures for their ability to degrade PCBs. At the same time, we also purified the bacteria from this mixed culture into their individual components to ask the question which ones had the PCB degradative competence and which were the best. When we did that, we found over 24 pure strains, each of which had a different ability to degrade PCBs, several of which were outstanding, and it is those that we are now continuing our studies with.
In the lab, they have singled out a number of genes which produce the enzymes that actually do the work of breaking down PCBs. When bacteria containing one or more of these genes were sprayed on a test site, a quarter of the PCBs were destroyed. But it took 4 months. Genetic engineering could improve this 1,000 times, the scientists believe. Although whether these gene-spliced bugs will ever be allowed outside the lab is another matter. When tankers started to go down, a young scientist thought there must be a better way to clean up after them. The storms that wrecked the tankers also wrecked the contraptions designed to prevent pollution. It was back to buckets and spades until the invention of the oil-eating bug.
And then get rid of the chloroform ether again.
Ananda Chakrabarti was the man who made the bug. But it took 5 years to establish his right to patent what was, in fact, the first living invention. Chakrabarty has been challenged every step of the way by people who think that what he does is dangerous and wrong. He does not use conventional gene splicing to create his cleanup bugs, but all the same, you won't find them in nature. Besides their main chromosome, many bacteria have extra ring-shaped molecules of DNA called plasmids. Each plasmid is composed of several genes. When bacteria mate, one can donate a copy of a plasmid to the other. This is what Chakrabarty exploits. To create a strain of bugs that can break down a complex chemical, he starts with one containing a suitable gene to begin the process. Other bugs are introduced with genes that will continue the job. Their surroundings and food supply are manipulated until the shuffling of plasmids produces a new strain. A strain in which all the useful genes are combined. The enzyme produced by oil-eating bugs is used to clean out pipes and tanks in the oil business. But the bugs themselves have never gone into action against pollution. In the lab, there are bugs that break down some of the nastiest chemicals known to man, but scientists are reluctant to let them out.
We don't want to do it because there are no clear-cut guidelines, and therefore I'm subject to lawsuits. These are usually nuisance lawsuits that people might bring, claiming that the microorganism I released might have somehow caused health problems for them and all that. So in general, we like to field test any new microorganism or product under supervision of the Environmental Protection Agency.
The career of another banned bug began here on the ski slopes. When nature lets us down, snowfalls can be manufactured thanks to bacteria called Pseudomonas syringae, also known as Ice Plus. Everybody knows that water freezes at 0, but not always. When all the Ice Plus are removed, it remains liquid at -6. Add some Ice Plus, and there's a snap freeze. Just 1 or 2 degrees of frost is enough to cost fruit growers millions of dollars a year. That's why a California company decided to wage war on these ice-making bacteria. They did it by creating another bug, in every way identical to Ice Plus, except they'd taken out the freezing gene. They called it Ice Minus. Strawberries sprayed with it survived exposure to frost, or at least the fridges in the lab. Legal battles raged to decide whether it was safe to test Ice Minus in the field. In the end, the inventors won the day. But the protesters saw its significance.
Earth first, no designer genes!
The first permitted release of gene-spliced organisms.
Genetically engineered organisms are very analogous to the introduction of exotic, non-native organisms into our ecosystems from all over the world. Most of them have fit in, some of them have died out, but a few of them have become tremendous pests, causing billions of dollars a year in damage. Kudzu vine in the south, Dutch elm disease in the north, chestnut blight, starlings, gypsy moth. These are organisms from other parts of the world that found a habitat, proliferated, and became pests. We can't get rid of them. Now imagine the scale here with genetic engineered organisms. Imagine introducing scores, then hundreds, and then into the next century, thousands of genetically engineered viruses and bacteria, plant strains and animal breeds, in massive commercial volume all over the world. Sheer statistics suggest that if only a small fraction of these organisms turn out to be pests, the damage could exceed in magnitude the damage we face with chemical pollutants, because these organisms, they're alive, they reproduce, and you can't get rid of them once they're in the environment.
In the case of gypsy moth, you're talking about something that is a natural component in and of itself, the ecosystem. Apparently, the predators and parasites that are necessary to keep populations of gypsy moth down are not present in sufficient numbers. In the case of Ice minus, all of the normal competitors, predators, parasites are in place because its enemies are the same as the enemies of the original strains. So it's not at all the same as introducing some kind of foreign organism into an ecosystem that's not prepared to deal with it.
At public hearings, the inventors explained that their Ice minus bugs work by simply occupying a few spaces that Ice plus would have taken. Others claim they are sowing the seeds of disaster.
You see, this particular bacteria is found all over the temperate regions of the world. Pseudomonas syringae, according to the researchers, and it's speculative, they think it blows into the upper atmosphere, triggering rain. It's an ice nucleating bacteria. AGS eliminated the genetic instructions that make the ice. That could have long-term effects on lack of rainfall or stopping rainfall patterns. Is it worth a few temperature degrees of benefit to frost retardation if we have a long-term impact on rainfall patterns? Again, it's only a small probability risk. But if this risk materializes, the problem could be irreversible.
The amount of risk assessment studies that AGS was required to do before they were issued a permit to do this test is unprecedented in the history of the development of microbial pesticides or in the history of the use of microbes in the environment. And this was because there was a lot of concern, not only public concern, but also concern within the regulatory agencies, but also because those of us at AGS who are involved in this thing are environmentally concerned, and there were a number of tests that we would have done regardless of whether or not they were required, because we want to make sure that that the field test would be safe.
Yeah, we are in a rather Kafkaesque situation here. There is no risk assessment science to judge the risk of any of these introductions. There is no database on how genes transfer in nature, how organisms compete, how they survive. So we're in a situation of the government saying it's regulating a technology, knowing there's no predictive ecology or risk assessment science by which to regulate it.
The arguments basically have been that somehow if a microorganism is genetically manipulated, it becomes somehow dangerous. Now that's a very weak and untenable argument because, researchers in the universities have been doing genetic manipulation for years. And as a routine procedure, we always isolate new type of microorganisms from nature. We change them genetically, and then we actually release them in the environment by dumping the whole thing in the laboratory sink. So, and there has absolutely been no incidence of any kind of hazard to the environment.
The debate continues. Californian strawberries are as prone to frosting as ever. The PCBs and dioxin in Luella Kenny's backyard may prevent her grandchildren playing there. Ananda Chakrabarti says it's a storm in a teacup. He is simply revving up evolution.
If we didn't do it now, nature would have done it over a period of 50 years. And what we are saying is that can we afford to wait for another 50 years for microorganisms to develop that would clean up the dioxins and the PCBs and the Agent Orange.
The pretty toadstools that grace the forest floor are the pearly kings and queens of the enormous family of fungi. For each one in the fall parade, 1,000 more work out of sight, spinning 1,000,000 miles of threads to feed themselves and bring about decay. Yeasts are part of this industrious family, which helps make bread, wine, beer, and smelly cheeses. If biotechnology has its way, their work is only just beginning. Fungi are not always friendly. When the Americans fought in the Pacific, they counted among their enemies, besides the heat, the damp, and the Japanese, a fungal fifth column. Uniforms, tents, and rifle slings disintegrated before their very eyes. It called for drastic measures.
In 1942, I believe, they sent Lawrence White, a fungal taxonomist, to go out to the tropics and go around the world and pick up samples of materials that had been degraded and bring them back with the idea that we would isolate the organisms and study them and see if we could find good ways to prevent this deterioration.
Mary Mandels and Alwyn Rees were the Army biologists who grew the fungi and compared their digestive habits.
When a fungus grows on cellulose, it doesn't eat the cellulose, so it has to secrete enzymes that digest it. The enzymes turn the cellulose into sugar, glucose, and then the fungus absorbs the sugar and grows on that sugar. So in the process, you destroy the cellulose and create a great deal of fungus, and you end up with a rotten, moldy piece of material. So for 10 or 15 years, Alwyn Rees and myself studied the fungi that grew on cellulose and studied the enzymes that they made.
They found it was the combined effect of two enzymes that made the sweet-toothed fungus such a prodigious converter of cellulose to sugar. In their excitement, the original purpose of the research was forgotten.
As we began to make these enzymes in larger and larger quantity, it would often occur to us that we shouldn't really be fighting these enzymes. We should be using them, because if you could take all this waste cellulose which is available and convert it into sugar, you would have a very useful process. And at first we thought of using the sugar as food. During the 60s, for example, there was a lot of talk of the world running out of food, hungry people. Well, you could produce single-cell protein very nicely on the sugar that you made for cellulose. You could also eat the glucose.
Old tabloids were now the favorite fare of the veteran fungus. But even the lab director's dedication to the project, anointing his breakfast pancakes with newspaper syrup, failed to get the team on the front page.
When the oil crisis came along in the 70s, we transferred our interest to making alcohol from the sugar, and that, I guess, is when we really went into high gear. That's when the Department of Energy began giving us considerable money. The laboratory grew. We began bringing in engineers. We were able to develop the process, and we built a pilot plant.
The power of the press, which had failed to feed the world in the 60s, evaded them yet again. This time, molecules were the problem. The enzyme cellulase was curiously shaped. Cellulose itself was inconveniently large.
If you want to think of cellulase as looking like a cigar, you can think of a piece of cellulose as looking like a basketball. So instead of two nice small soluble molecules coming together and complexing, you have this basketball with a cigar trying to attach itself to the surface. And the hydrolysis is very difficult. Therefore, it requires a tremendous amount of enzyme to make sugar from cellulose.
Right up until their retirement, Mandels and Rees continued to refine the process, juggling the science, the engineering, and the economics. There were many small steps forward, but never a big enough leap.
The enzyme requirement made the process too expensive to be practical today, and that's why it hasn't gone commercially yet, but it will someday.
If the Army process had worked, the end result would be ethanol. Today, ethanol is in the spotlight. There is pressure on oil companies to put ethanol in gas instead of poisonous lead.
Ethanol is a renewable resource. It's made from living things that grow year after year, and that's its main advantage. It also contains oxygen, which makes the exhaust emissions much cleaner. In the United States right now, there's quite a bit of activity in actually mandating the use of oxygenated components in gasoline as a way to meet the ambient air quality standards in a number of cities.
Apart from the new name on the outside, this ethanol plant in Manitoba has changed little from the days when it made whiskey. Like any spirit, ethanol is distilled from a weaker alcoholic brew. Here, the raw material is grain. It's mashed with water, and then yeast is added. In the fermenters, it takes the yeast a couple of days to convert the grain into alcohol for distillation. The plant can make 2 million gallons of ethanol a year from grain. But a fungus from the South Pacific could change all that. Not for the first time, Ed Malik is summoned from a warm bed by Trichoderma reesii. Such is the devotion of a team who, like the Army scientists at Natick, believe that wood can be converted into ethanol by the battling fungus.
There have been a lot of changes in trichoderma in terms of improving it. Since the mid-70s, there's been a roughly 100-fold improvement in the overall productivity of the microorganisms. That's come through genetic changes to the microorganism and very substantial improvements in the fermentation technology, the actual growing of the microorganism.
It's the digestive juices, the enzymes, that are still the key. The team find they get the most by keeping the fungus lean and mean.
Essentially, he goes through life thinking he's about to starve to death. And his response to that is to be pumping out large amounts of this enzyme. And at the end of a typical run, we do starve him to death, kill him off, and collect all the enzyme. On the count of improving the performance of the enzyme on wood, the keys are improving the pretreatment so that you have a huge amount of accessible surface area, probably a factor of between 5 and 10 more than what they ever had to work with at Natick. And the second part is around the hydrolysis technology. That's the step where the enzymes break the wood down to sugar.
Iogen won't reveal too much about their process. If they become the first to make it work on a commercial scale, it is hard to overestimate its worth. But first of all, steam and pressure puff up wood chips like breakfast cereal. Then the enzymes are added. Finally, the results are filtered. It's the sugar syrup that passes through the filter that will be turned into ethanol. But what happens to the brown cake that's left will decide whether the process is a moneymaker. The brown stuff is called lignin. Lignin binds together the cellulose fibers that make up wood. It's a nuisance because it gets in the way of the enzyme attack. Iogen are turning the problem to their advantage by experimenting with lignin as a glue. If they can make it stick, the income from this byproduct will make the whole process economic. The scientists say we'll see that within 2 years. If they're right, the days of making fuel from food will be numbered. We'll be re-entering the age of fuel from forests.
The Brazilians have a vast alcohol fuels industry based on sugar. Its success really goes up and down with the price of crude oil and the price of sugar on the world markets. When we're talking about making ethanol from wood, we're talking about a much stabler commodity, something that really doesn't have the same kind of high-value alternative uses.
But trees have more than a balance sheet value. How will we gauge the impact of this new industry on the environment?
When things come to the point of extremely large-scale utilization of biomass, we won't be denuding the earth so much as building forests around the earth. It's not going to look like a strip-mined coal area. There'll be lines and lines of farms, presumably new farmers and new business. So in fact, the world will be greener as a result of this. But obviously in the areas in which people do this, there'll be a change. In Canada, for example, a 14-mile wide strip across the country would grow enough trees on a steady basis to produce all the gasoline requirements for the country.
The picture is not so rosy for more densely populated countries. Putting a tree in every British tank would mean foresting an area larger than Scotland. Perhaps this is why even Foodie is not predicting an overnight success.
There'll be billions of dollars invested before ethanol makes up even as much as 10% of the total U.S. gasoline requirements. I would think that one would have to wait 10 or 15 years at best before you'll see that very significant industry in place.
Lignin is very versatile. As well as glue, it helps to make rubber, roads, ink, chemicals, and ice cream. Most vanilla flavoring is made from lignin, but now it has a challenger. Soon, all the world's vanilla requirements could be satisfied by a handful of plants in greenhouses. It's the very latest in biotechnology, but its inventor says it's perfectly natural.
It's natural in the sense that it's made from a natural material, which is the plant cell, the same plant that makes the vanilla bean from which the vanilla flavor is usually extracted. It's the cells are nurtured with natural ingredients. They're nurtured with air, oxygen, sugar, and we produce the flavor. So in that sense, and in a very real sense, in a regulatory sense, it meets the characteristic of the qualifications for natural.
Moshe says vanilla orchids are sickly and prone to disease, so only those which pass a stringent fitness test are used. Most of the material is thrown away, but a few cells are selected to grow in liquid cultures. The cells are mixed with warm plastic and dropped in water, the way that shot was made in days gone by. Each spongy ball contains up to 1 million cells, all able to convert air and sugar into pure vanilla. It's clever. But could these miniature factories manned by cells soon be causing problems for people? Many of the steps forward made by technology have turned out to be steps back for the third world. Nylon has replaced natural fibers. Many things besides sugar now satisfy our sweet tooth. Our boundless ingenuity has reduced the market for traditional products. Most natural vanilla comes from Madagascar, earning 10% of its income. The Malagasy cannot afford to ignore this innovation. Can we?
Certainly it's true where we now can see that through tissue culture process, the crop will move from Madagascar and 67,000 farmers who grow the crop in Madagascar to the United States and factory production in the United States. And that is very much bad news for producers in Madagascar. But I'm not really convinced that in the longer run it's going to be good news for people who like to eat high-quality vanilla either. I'm not sure that the product, once it is controlled that way, once it's in the hands of one or two major companies who will have the market, where the quality will stay that way. Once you've lost the alternative, the natural alternative, and you can only take the laboratory product, I think consumers will ultimately lose out.
Our quality will be equivalent to the natural vanilla from the vanilla extract and far superior to that of the synthetic. So we would like the natural vanilla bean extract to remain. More importantly, in this particular case, we do not intend to displace it. We intend to displace part of the synthetic market, a tiny part of the synthetic market. 1 or 2% displacement of the synthetic market doubles the natural vanilla market. So the natural vanilla market will remain. The farmer, whether it's the Madagascan farmer or the farmer in Ghana, really doesn't have all that good a life. Agriculture isn't doing all that much for him. What we see happening is there have to be some intermediate, very temporal dislocations when you change a technology. There's the same dislocations when you change a crop. We see the worker moving from plowing the field to making microchips, from harvesting the wheat to making designer clothes. The displacement is there. Far less than a 5-year drought on the farmer. Far less than a 5-year drought. You don't get 5-year droughts in the computer industry. You might get a year drought. So I don't discount the importance of it. But net, it certainly is not harming humanity.
Most things that can be grown in fields can be grown in factories, in efficient, controllable conditions, wherever you want. Right now it's pretty expensive. Only a few drugs and high-value colorings and flavorings are produced this way. But the costs are reducing every year.
It's a long way still from the price of wheat. There are very few people who are going to work as cheap as farmers right now for our cereals. But the price is constantly dropping. It's not a technology which I think we can safely say is just going to be used for exotic plants. It's going to finally, someday, be theoretically, economically viable for most major crops.
It's not going to do it because economically, the price of a phyto-produced product will always be in the dollars and many dollars per pound. Corn and wheat and sugar are in the cents per pound. So where is it going? It's going more and more for the value-added additives. It's going to go to antioxidants. So we'll have natural antioxidants, so the natural soybean oil or the natural peanut will have more shelf stability. It's going to go to natural pigments, so the naturally pale green orange will look naturally bright orange with natural pigment. That's where it's going to go. It's going to go to enhance the quality of life rather than just to feed the belly.
All right. Okay. So, Jenny, you just have to be ready.
Biotechnology's true believers say our children will inherit a different world. The dark, satanic mills of the last industrial revolution will be swept away by palaces of glass and stainless steel, in which cells and microorganisms will be the workers. Industry is already discovering what a willing workforce they are. Start with just a few. Not any old bacteria, but ones in which you've spliced a gene which produces a particular protein. They multiply so fast that a teaspoonful of bacteria would end up weighing more than the Earth in a couple of days if conditions were perfect. Make sure to keep them warm and feed them well. Bacteria are never happier than when they're agitated. Soon, billions and billions of bugs will be drooling those precious proteins into the microbial brew for you to remove with filters. That's how a factory in Wisconsin can produce enough food enzyme to fill an 8-story tank from a tiny test tube. But, like all something-for-nothing deals, there's a catch. All goes well if you ask the bugs to produce variants of proteins they know. But if the gene you splice into them is the recipe for a protein they don't know, they put in all the right ingredients and turn it out the wrong shape.
In the first place, many microorganisms can't fold proteins correctly, and it can be extremely complicated to take that protein and have to reactivate it after the bug has made it. I've heard it likened to trying to turn a meringue back into the egg white from which it originated. Whereas mammalian cells will secrete these natural proteins in their biologically active natural form.
That's why Britain's biotech flagship does not employ the ignorant bug. The pharmaceutical proteins manufactured by Celtech are produced by shifts of mouse and hamster cells.
We insert into that mammalian cell the DNA, the genes that are required to make protein that we're interested in. We then put those cells in their nutrient medium into a big stainless steel vat, a fermenter, much like the sort of vats that you might use for growing microorganisms. The cells secrete the product into the culture supernatant. We can then recover those proteins from that culture supernatant.
Again, it's easier said than done. And though unwanted, the redundant microbes are refusing to go quietly.
It's a process which is easily contaminated with microorganisms if you don't take extreme precautions to avoid microbial contamination. So there have been some considerable technical difficulties to overcome in designing and engineering processes which can be run aseptically. And one of the great achievements that we see is that those processes have been achieved. We do now have industrial processes where we can grow mammalian cells on a very large scale and make kilograms of these products with sterile processes.
But the protein producers of the future may not come in glass and stainless steel at all. This flock of sheep will not end up as lamb chops. It's been bred for a very special purpose.
The notion in general is, can we use animals as bioreactors rather than the conventional types of bioreactors that we're more used to these days, such as bacteria and tissue culture lines.
The gene for a human protein, a blood-clotting agent vital to hemophiliacs, has been spliced into the sheep. It is hoped the protein will be secreted in the sheep's milk.
And the way we do that is to physically micro-inject one-cell fertilized eggs of sheep just after they've been ovulated and fertilized, and then reimplant those micro-injected eggs back into recipient animals.
The human gene is now a permanent part of these sheep and of their offspring. If their milk contains blood factor 9, it'll be a breakthrough for biotechnology with natural advantages.
Tissue culture systems are difficult, they're expensive to run, there are problems in scaling up. You have a sheep, it by and large it lives on grass, and it scales up in what we might say a traditional manner to give us a flock.
next time, when the wrong choice of entree can put lives at risk. How the same reaction can be used to detect cancer and even to treat it. See exactly how AIDS mounts an attack, while biotechnology prepares to fight back. And we look at the genes which cause cancer. Could we be vaccinated against it? Join the life revolution and find out.